Hillary, that's actually right above. Hi, John.
Ready to go?
What time is it?
Okay. So let's see, origin stories. Story. Bob Warner and I met Zoe, I think when she was a sophomore in the Evolutionary Medicine College of Creative Studies class. And both of us realized that, she had opinions, she had thoughts, she had a mouth on her, and we would know her for quite a while, as it turns out. There. So that's the origin. I've just confirmed it with Zoe. But what I really first want to talk about is baseball. So baseball, there's a characteristic. It's called a five-tool player. There are very few of them. Willie Mays is the archetype. It's a player who can hit for average, hit for distance, catch balls, run bases, throw balls, do all of the things. It's rare in baseball and rare to be exceptional. And I would say, as a zoologist, as an ecologist, Zoe is at least a five-tool player. The thesis she undertook, working to resolve the food web of the Rocky Intertidal Zone, was a massive project. To put it in perspective, when we did our well-funded research on the estuaries of California and Baja California, to construct the food webs, we had a team of over 30 people, and it was very well funded. There's a couple of hundred, less than 200 species in all three estuaries we worked on. There's well over 1,000 species in the Rocky Intertidal Zone, and Zoe did this research on her own with minimal funding. So that requires a tremendous amount of data, both from her observations and collections in the field and from the literature. And I will just say every one of those species required multiple thoughts about what they're doing, what their role is, who consumes them, what do they consume, and a whole host of other bits of information. So there's a ton of information that Zoe had to analyze, compile, organize. And so to do that, a second major skill was leadership. She developed a team of mostly undergraduate research assistants who really did quality work with her. She has engaged this complex, massive topic into theoretical aspects, because there's some really important novelties there. Beyond just the research, she's a stunningly good lecturer. I I pride myself in my lecturing ability in courses, but once she started co-instructing with me, boy, I struggled to keep up, and it fed my competitive juices. That was good there. But the reviews that she had and her teaching, my teaching is classical. Hers is, I think there's a word for it, but I'll call it hyper modern there. And so very different style and very effective. Leadership, during the troubling times, pre-COVID, the DEI period, Zoe was an important leader of our graduate students, and led not with just, it wasn't just being out there in front, it was organizing documents and doing the important work that you don't get quick credit for. And so lastly, most of our theses in our department don't have a direct management aspect. Zoe's does, and so it plays a role, it will play a role in how the coastal environment is managed. And of course, can we say your position? Zoe has been hired by the National Park Service in a really important position to do just exactly that sort of thing. Anyway, I totally look forward to hearing what she has to say.
Thanks, Armand. Wow, okay, I can follow that. Fine. Thank you guys so much for coming. It's amazing to see everybody here and amazing to have everyone together for the culmination of 6+ years of work. I'm going to try to go over it in an hour. We might go a little bit over, but it's, 6 years of work, so we'll see how it goes. The title of my dissertation is Connected by Tides, Missing Links in California's Coastal Ecotone. So I'm going to start off by introducing you to this incredible ecosystem that's right in our backyard, the intertidal ecotone. Sorry for those of you on Zoom if the intertidal is not in your backyard, you're missing out. There are 3 chapters of my dissertation that deal with the intertidal zone. First, building a food web for the intertidal zone, Armin just introduced. Second, exploring the role of infectious processes in the intertidal. And third, exploring the role of cross-ecosystem processes in the intertidal. So we're going to start by introducing you to the rocky intertidal zone. Myself and the kids call this the Riz. If you're not familiar, Riz is a slang term that means charisma. I think the rocky intertidal has a lot of charisma, but you'll hear me refer to it interchangeably as the rocky intertidal or the Riz. Now, when people think of the intertidal zone, a lot of times they think about tide pooling. Most of you have probably been tide pooling. If you haven't, go. But the intertidal is a lot more than just tide pools. It is a pretty narrow strip of habitat that spans from the supralittoral, so above the highest high tide, to the sublittoral, below the lowest high tide. And it contains a wide variety of habitats and environmental, all sorts of environmental parameters change within the rocky intertidal zone. There's also a whole bunch of microhabitats contained within the rocky intertidal zone. So again, not just tide pools, but we've also got bedrock shelves, algae mats, mussel clumps, surge channels, reef, etc. So all sorts of microhabitats that lead to a rich diversity of organisms. So the rocky intertidal is one of the most biodiverse ecosystems on the planet, especially here on the West Coast, where we have our rich upwelling that provides a whole bunch of nutrients and transports a bunch of larvae into the rocky intertidal. So in short, a lot of ecosystems, or a lot of organisms, a lot of really cool microhabitats. It's also pretty easy to get to the rocky intertidal. You can just walk down to the beach, find some rocks, and start doing research. So people have been doing research there for decades. And as a result, on the West Coast of North America, there has been some of the preliminary evidence for a lot of foundational theories that you learn about in Intro to Ecology. For example, the keystone species hypothesis, the intermediate disturbance hypothesis, and there's evidence for interspecific competition. All of these were first observed in the California, not the California rocky intertidal, but the West Coast rocky intertidal. Oh, yeah. Hold on, it's gonna take me a second to get all the way over to the screen and then hit OK. Oh yeah, that just goes right over all my cool titles, doesn't it? What a bummer. Thanks, Bob. Okay, so rocky intertidal, we know her well. Now I got to get back to this screen. Okay, so because we've been working in the rocky intertidal for so long, there's a very rich depth of knowledge that has allowed us to not only develop foundational ecological theories, but also develop these really detailed lists of all of the organisms that live in the rocky intertidal, encompassing all of the phyla that live there. And because of this, we can do really effective long-term monitoring in the rocky intertidal. So the intertidal on the West Coast is monitored by these three organizations: PISCO, which is the Partnership for Interdisciplinary Studies of Coastal Oceans; Marine, which is the Multi-Agency Rocky Intertidal Network; and the Santa Barbara Coastal Long-Term Ecological Research Projects. So in short, we have a very rich history of studying the rocky intertidal. We understand it really, really well. I am one of the people who understands it really, really well. I've been working in the rocky intertidal since 1993. Only recently did I start to take my studies to the next level and take them really seriously, thus beginning this PhD, which I'm going to walk you through now. So we'll start by looking at the food web for the rocky intertidal. Now, what is a food web? Food web is a collection of feeding interactions between species that represents a system. Now, this figure looks kind of abstract, but we can make it a little bit more clear by adding some animals that we all recognize. So the food web takes organisms and it relates them to each other, connects them to each other based on who eats who. So here we have the bird who eats the fish, and the fish who eats the crab, and the limpet, and the bird also eats chitin, mussel, and barnacle. Now, why build a food web? What's the point of this? There's actually a lot of really interesting reasons to build a food web. First, we can use food webs to relate discrete interactions between two species to an entire ecosystem and to ecosystem-wide patterns. Second, we can use food webs to understand the movement of energy through a system from the lowest trophic levels, the primary producers, to the top predators at the highest trophic levels. We can also use food webs to predict indirect effects. So trophic cascades, for example, or competitive release. Give you an example here. If this bird at the top of this food web were to go extinct, what would happen? We could use the food web to predict that. Because the bird eats the fish, and all of a sudden the fish is released from predation pressure by the bird, fish populations increase, then they could maybe depress the populations of their food items, therefore releasing the competitors with those food items so their populations can grow. So essentially we can use these connections to predict what might happen if species go extinct. We can also use food webs to understand on an ecosystem scale the relative contribution of functional groups. So for example, here we can classify organisms as carnivores, grazers or herbivores, and filter feeders, and we can kind of understand the relative contribution of these different functional groups to the whole ecosystem. Now, what this allows us to do is understand, again, on the ecosystem scale, ecosystem services, so things that the ecosystem provides for us, human beings, ecosystem functions, so how the ecosystem works, and especially in our changing climate, unfortunately, resilience to change. So how that might work is, say, for example, half of our rocky intertidal food web goes extinct. Because we have redundancy in our functional groups, we now understand using our food web that we might still have grazers, filter feeders, and carnivores, even if half of the food web goes extinct. So food webs can help us sort of predict outcomes of change in ecosystems. Now, other people have attempted to build food webs for the West Coast rocky intertidal zone, but those food webs have been kind of constrained and not as taxonomically resolved as we'd like them to be. Now, what taxonomically resolved means is resolved all the way to the lowest taxonomic unit, which is species. So a lot of food webs will clump their organisms together into groups, and that sort of dilutes the resolution of a food web. So this food web for Tatoosh Island in Washington was really wonderful and very well thought out, but it wasn't super highly taxonomically resolved. And what's more is it only represented one chunk of the rocky intertidal. So it only represented the mussel bed zone, which I've outlined here in blue, while neglecting the rest of the habitats within the rocky intertidal. So basically, what we need is a taxonomically resolved food web that represents the whole rocky intertidal zone. And that's where I came in. So before I walk you through the food web, I'm going to tell you how I did it. How do we build a food web? Let's break it down into its component parts. So food web is made of two parts: nodes, which usually represent individual species or sometimes, again, functional groups that would that all consume and are consumed in the same way. And the way that we did this, and by we, I mean me and a very large team of undergraduates, is we use these two books that have lists of species that are very taxonomically well resolved, as well as long-term monitoring data from the multi-agency Rocky Intertidal Network to build a really comprehensive list for the entire Rocky Intertidal, encompassing all of the habitats within the Rocky Intertidal. Next up, we build the links. So links are the feeding interactions between the nodes, and they are these physical lines that connect the nodes to each other. And this was the hard part. So step one of building all the links list was to track down and read over 1,000 papers. My team and I put together a list of about 500 papers that contained information about feeding interactions, but to find those 500 papers, we had to read twice as many. We got really, really good at Googling, which is a skill that most PhD students gain in this day and age. Step two, after we were done using the literature and Google to build our links list, was to collect and dissect organisms to further identify interactions between species in the Rocky Intertidal Zone. Now, I put these 3 pictures of me and 1 picture of Lauren in the Rocky Intertidal to illustrate the adventures we got up to, but it wasn't just me. I had a massive team of undergraduates and postgraduates and some high school students help me out with this effort, and I just want to take a minute to thank them all. And I'm going to do, sorry, Jaden, a shout out to Jaden Orley, because she has been here since day 1 of this project and is still here right now and has helped me every step of the way, from 16-year-old high school student to co-author and collaborator. And I'm just so, so proud of her. She's been instrumental to this project and could not have done it without you. So as a team together, we did over 500 dissections of fish, snails, crabs, worms, etc. And while we were doing those dissections, we were looking in the guts of these organisms for their gut contents, as well as for their parasites. So identifying interactions with other organisms using dissections. Then once we had put all of that together, it was time to construct what we call a network graph. So a network graph relates the organisms together by connecting them with these lines, right? And one of the things that's really important to note is that we put the lowest trophic level organisms, so these are our primary producers and things like dead stuff and detritus, at the bottom of the food web. And then as you go up, you go higher in trophic levels, such that the top predators are at the top of the food web. Okay, so this image is a very early draft of the food web. When we were all said and done, this is what our food web looks like. It's not very informative, but that's okay. It informs you that this was a lot of hard work. One of the really cool things about this food web is that it's actually the biggest food web that has been assembled so far. I would say published, but we're not there yet. We're so close. The food web is comprised of 1,845 nodes, 1,600 plus of which are resolved to species. So that's a very big deal to get all of those nodes resolved down to species. Again, most people clump organisms together into functional groups. And between those species, we had over 13,000 trophic links. Now, every single one of those links is a row in a data sheet, so just keep that in mind. Just some fun, like baseball card facts about the food web that I thought I would share with you guys. First, connectance is a metric that people use to describe food web complexity. Now, what connectance does is it tells you how many links out of all possible links actually exist in the food web. So our food web is 0.5% connected, which basically means that of all possible links, we have actually realized 0.5% of the links. So it's not super connected as far as food webs go. A 100% connected food web would mean every single organism interacts with every single other organism. Now, another way that we can look at food web complexity and like sort of the rate of interactions between organisms is by looking at average degree. So degree describes the number of links per species, and on average, every single organism interacts with about 18, 19 other organisms, and that's including being eaten or eating things. But there was a wide range. A lot of organisms only interacted with one other organism, and the most connected species in the web interacted with like 150 plus other organisms. So some of the most connected species in our web include the shorebirds, and they're up there because they eat everything. And then one of the other most connected organisms in the web was Cancer anthonii, which is the Rocky Intertidal yellow crab. And they not only eat everything, but they're also eaten by everything. So super well connected within the web. most people think of, instead of food webs, they think of food chains. And within food webs, there are a lot of food chains of varying lengths. In this food web, the average food chain was about 4 organisms long. So this example of a food chain here goes from algae, which is then eaten by a limpet, which is eaten by a fish, which is then eaten by a bird. So on average, this is about what our food chains within the food web looked like. Now, I'll remind you really quick, one of the reasons to build a food web is to understand the relative contribution of different functional groups. And so we'll go through that really quick. And within this food web, about 14% of the organisms were predators, about 7% were omnivores, 10% grazers, 4% primary producers, so algae and plants. And then the majority of the organisms in this food web were filter feeders, which definitely makes sense. You're in a habitat that has a lot of moving water. And in order to survive in that habitat, you should probably hold really still because of all of the moving water. Best way to obtain food, if you're holding still, is to filter feed, especially if you're in the water. So of the predators, here are some fun examples. Got gulls, octopus, and sea stars. Omnivores include, again, crabs and shrimp, a lot of other crustaceans. Of our filter feeders, we've got mussels, barnacles, sponges. Grazers include a lot of mollusks, limpets, chitons, and I put an abalone here because they're one of my favorite critters in the food web. And then of our primary producers, we have 2 actual flowering plants, as well as green algae, red algae, and brown algae. Now, if you know me, you know that I care about all those guys, but what I really care about is the parasites. Now, this is a functional group that is relatively understudied, underappreciated. Most people think they're icky, but most of you who know me, you know, I don't think they're icky. So, of course, I spent an entire chapter of my dissertation looking at the role of parasites in the rocky intertidal. All right, so why parasites? Turns out parasites are actually really important components of food webs. They've actually been referred to as the ultimate missing links by our very own Kevin Lafferty. Thanks, Kevin, for giving me a beautiful title I could put on a slide. And the reason that they are the ultimate missing links, because they're missing from a lot of representations of food webs, are because they are just another type of consumer interaction. So just like predation, parasites consume their hosts, although on a smaller scale. They act almost effectively like predators, and we shouldn't just leave out a type of consumer interaction because they're small, hard to see, and people think they're gross. Furthermore, sorry, everyone has parasites. So probably not you guys because you live in the Western country where we eliminate parasites, but in the rocky intertidal and elsewhere on the globe, almost every species of organism on the planet has a species of parasite associated with it. So seems silly to leave them out of food webs. Now, on top of that, they have important ecological properties. I'm going to walk you really quick through a fun fact about a lot of parasites. Is a lot of parasites have complex life cycles. So think like a butterfly that goes, you know, egg, caterpillar, pupa, butterfly. Parasites are really similar in that they go through multiple stages. And a lot of times what that requires is that they have multiple hosts, sometimes of different species. So this is an example life cycle of what's called a trematode worm. And now trematode worms start their lives out as eggs pooped out of birds or other vertebrates. But in this example, we're going to use birds. And this bird host, because it's doing the pooping out of the eggs, is referred to as the definitive host. I need you to remember that word. After it is pooped out of the bird as an egg, the trematode worm enters into a mollusk. And while in that mollusk, it becomes the next stage, which is called parthenite, which is not a word that you need to remember. But you do need to remember that the hosts where it is a larva, those are called the intermediate hosts. So sometimes there's only one intermediate host. In this example, there's 2. We go from the limpet to a fish. And then in order to get from the fish back to the bird, the fish needs to be consumed in a really important process called trophic transmission, another word that you need to remember. So because parasites require movement from one host to another, this specific type of parasite, in order to complete their life cycle, and that movement requires being eaten, they actually can affect the outcomes and the strength of predator-prey relationships, because the parasite wants to get from the intermediate host to the definitive host, and it's going to manipulate the behavior of its host in order to get from intermediate to definitive host. So this can result in what would otherwise be a normal predator-prey interaction being a lot stronger or a lot more intense. Now, parasite presence, because of how they interact with different organisms throughout their life cycle, can actually indicate ecosystem health and even restoration success if you look for the right parasites. Now, the reason that works is because, let's say you remove the bird from the ecosystem because it's been scared away by people, or the habitat isn't suitable enough for it, or it's gone extinct, or whatever. All of a sudden, this parasite life cycle breaks down and the parasites disappear from the system. So if you have parasites missing from a system, sometimes that can help indicate that that system isn't exactly intact. So one of the things that has come of this pattern is that high host diversity, so potential host diversity, is actually related to parasite diversity. And again, a very biodiverse parasite community can indicate a healthy, biodiverse, free-living community. Now, this exists outside of just the single ecosystem scale, and it actually exists if you look on the ecosystem to ecosystem sort of biosphere scale. So this figure relates food webs. We have all of the food webs that have parasites in them on this figure. So the food web size is down here at the bottom, and size means the number of species within a food web. So this is kind of a metric for diversity. And then over here on the vertical axis is percent parasites. So basically what we see when we plot all of these food webs onto this figure is that the higher host biodiversity that you have, the more parasites you have in the system, the higher the percentage of parasites is in that system. Now, I want to highlight two really important food webs on this figure that are local to our area, the Carpinteria Salt Marsh and the Santa Barbara Kelp Forest. Now, these were done just in the last 20 years. These food webs were constructed, and they have pretty high free living diversity and extremely high parasite biodiversity, especially the Santa Barbara Kelp Forest. Really high biodiversity, both free living and parasites. Now, these two systems are connected to the California Rocky Intertidal, not just physically, they're not just in the same place and connected by water, but they also actually share species. They share a subset of species with each other that sometimes move from ecosystem to ecosystem throughout a single lifetime. So basically, these organisms moving between these really parasite-rich systems could be moving parasites around between the systems. Now, if we look at all of these together, we see that in the salt marsh estuary, we have just over 100 species. 35% of those are parasites. In the kelp forest, we have a little over 1,000 species, and almost half of those are parasites. The Rocky Intertidal, we have over 1,800 species, so probably a lot of parasites, right? Well, we found of our food web, which had just over 1,800 species, 1,509 free-living species, and 336 parasitic species. Seems like a lot. About 18% of the species are parasitic, which translates to about 5% of all of the links being parasitic interactions. That's actually not that many, especially when you start to compare it to other food webs. So we have these other two food webs that are local. Remember, they're connected to the Rocky Intertidal. They have the same species kind of moving around between these habitats. They have way more parasites comparatively, respectively, than the Rocky Intertidal does. So that Rocky Intertidal has more potential host species than any other food web that we've assembled, and it has fewer parasites than almost all, except for this one mudflat in New Zealand that Armin has informed me is like 20 species, and it's not that good of a food web. So what about these missing parasites? Is this a feature of the Rocky Intertidal, or is it a bug? Did we mess something up? We came up with some hypotheses while we were thinking about this. The first one is that maybe the Rocky Intertidal is just too rough. Maybe it's just too dynamic of an ecosystem. There's a lot of wave action. There's a tidal movement every single day that goes from washing machine fully underwater to fully exposed to the air and the sun twice a day at high tide and at low tide. On top of that, there's massive temperature swings during the day. I don't know if anybody heard about in Washington a couple of years ago during the heat wave when Rocky Intertidal animals literally boiled alive in their shells. So that's the kind of things that not just the free-living organisms, but the parasites have to deal with. And it might just be that it's too hard for parasites to propagate and survive in order to get to another host in this environment. Now, there's some evidence for this hypothesis from other research. A good friend of mine, Anai Novoa, who did her PhD at UC San Diego at Scripps Institute, did similar research to mine, and she found that the higher up in elevation she went into the Rocky Intertidal, the more parasites there were. Now, higher up in elevation in the Rocky Intertidal, you have less wave action. It's a little bit more stable. It's really hot and really dry and really salty, but it's a little bit more stable. It's kind of the same unless there's a really, really high tide, right? So maybe in these higher tide pools, there is enough stability to support a higher richness of parasites, but in the lower tide pools, there isn't enough stability to support parasite communities. So that's just one hypothesis. Another hypothesis that we had that might not be mutually exclusive is that the vertebrate definitive host, remember that word definitive host, okay? They are really important for the life cycle of the parasite. If they go away, the life cycle breaks down. So if they are not spending their whole lives or even their whole day in the rocky intertidal, maybe they're not around enough to contribute these parasite eggs to the system, to contribute to this, the life cycle of parasites. In the rocky intertidal, most vertebrates, birds, sharks, large fish, marine mammals, are transient visitors to the rocky intertidal. So they come in at either high tide or low tide, and then they leave. Again, they might just be too ephemeral to be depositing parasite propagules into the system. If this was true, we would expect that there are fewer complex life cycle parasites, these parasites that require multiple hosts to complete their life cycle. And that is actually kind of what we see. So when we compare our system to the other two systems in California, the ones that they're, you know, arguably the most similar to, we see a smaller contribution of these parasites that require more than one host. So here on this figures, we have single host parasites in this orange-yellow color, multiple host complex lifestyle parasites in the red, and free living organisms in the green. So when you compare the systems to each other, no matter where you look, complex life cycle parasites make up a smaller amount of a smaller proportion of the community than in the other two systems in our area. Now, I will show you just for fun, because I like pictures of parasites, some of the parasites that we did see. So again, we saw a lot fewer of the complex life cycle parasites, which are the trematodes and the tapeworms, and another really fun kind of worm called the acanthocephalin, which we actually didn't find that many of. And then there's the parasites that only require one host. So for example, fish copepods, parasitic barnacles, this is like my favorite picture that's ever been taken, and other sort of monogene single host parasites. We found a lot more of those than we found the complex life cycle parasites. And there's some evidence from other intertidal systems that one of the most important aspects of a community that's rich in parasites is that it has a rich community of these definitive hosts, these vertebrate definitive hosts, both in small space and sort of on the ecosystem scale. So you need these definitive hosts in order to maintain high parasite biodiversity, and it might just be that we don't have that in the rocky intertidal. Now, finally, and again, not mutually exclusive, we thought maybe this habitat has just been degraded for far too long to support parasites. Remember, we can use parasites to indicate ecosystem health, and the reverse is also true. If there are fewer parasites, maybe the ecosystem is not as intact. The rocky intertidal on the west coast of North America has been exploited for about 10,000 years by humans. So we're not talking about contemporary exploitation, we're talking about historical exploitation. So what that looks like is not just overfishing of our nearshore fish communities, but fishing down the largest organisms in the food web. And because there's been this long, long history of overfishing and overexploitation of our coastal systems, we see a simplification of food webs. So I'm going to call out here one of my favorite extinct critters, the stellar sea cow, which used to be pretty abundant on the west coast, but was hunted into extinction, all the way into extinction by indigenous peoples as well as white settlers as they came in. It's entirely possible that this big vertebrate organism had a bunch of parasites, and when it went extinct, so did all of its parasites. We're speculating here. We're not sure. These are all just hypotheses that could all be interacting together to contribute to the lack of parasites in the system that we see. Again, if hosts are being fished out at multiple levels in this food web, it's entirely possible that their parasites are being fished out as well. So to wrap up this chapter, infectious processes in the RIS, they are potentially absent. It's also entirely possible that they're under-researched or overlooked. They are, at least in terms of this food web, they are absent. And it's unclear whether or not that absence is a signal of a degraded system or a naturally unsuitable habitat for parasites. Okay, I don't have a fancy transition here. We're just going to move on to the third chapter. So finally, once I was done with the food web, I decided to look into cross ecosystem processing, and we'll talk about what that is. Up until now, we've been talking about the rocky intertidal zone, right? But the rocky intertidal zone is not a standalone habitat on an island. It's connected to other habitats. And one of the really cool things about the intertidal zone is that it's full of a whole bunch of resources, and animals can move across ecosystem boundaries to obtain those resources, specifically mammals. Now, maritime mammals, which are terrestrial mammals that come down into the intertidal zone to do stuff, they're a global phenomenon. They've been seen on every single continent on the globe except for Antarctica, and they're reported to actually forage in the intertidal. So they're not just coming down there to hang out, they're actually eating stuff. And again, every continent, every coastline except for those on Antarctica. Now, this has probably dramatically changed from historical maritime mammal abundance and activity, right? We're not going to see a whale fall like this result in a bunch of grizzlies coming down to the beach to scavenge on that whale fall because A, we don't have grizzlies, and B, what usually happens to whale falls is they get dragged out to sea and sunk or blown up. So this process doesn't exist as much as it probably used to. Also, because most of California looks a lot like this. People like to build their homes and do their fun stuff all the way up to the edge of the marine habitat. And what happens when there's human development in the way of animal movement is they don't move. One of the other things that's pretty prevalent on the coast of California is the 101 freeway. And there's extremely high rates of wildlife vehicle conflict on the 101, especially in our area. So this is Point Conception right here. This is Santa Barbara. It's possible that that extremely high wildlife vehicle conflict is because wildlife are trying to get across the 101 from the terrestrial habitat to the marine habitat. Now, we've actually received some anecdotal reports before I started my research of mammals active on beaches in the more remote parts of Santa Barbara County. So the Gaviota Coast area, which is kind of west of Elwood, west of campus, over like towards Point Conception. The interesting thing about that area is that it's not super well developed. It's made up of Hollister Ranch, Jack and Laura Dangermont Reserve, and Vandenberg Space Force Base, and there's not a lot of human development on the coastline. As a result, there's really high habitat connectivity compared to the rest of the South Coast. The 101 isn't blocking access to the ocean. We don't have Santa Barbara or LA physically impeding animals from moving across habitat boundaries. Furthermore, this part of the coastline is actually protected both by the Nature Conservancy and to a lesser extent by the Space Force Base. So this is a great place to answer this question. Are maritime mammals in California a missing link? And because no one's actually tried to answer this question before, I got to answer 5 very basic questions. Who, what, where, when, and why. Now we can expand those out a little bit. First, we asked who is using the beach habitat? What mammals are using the beach habitat? What are they doing there? Where are they active on the beaches? When are they active on the beaches? And of course, why does this matter? So we started a project called Project Ecotone. And that project involved using 40 motion sensor camera traps that we set out from April to December 2022. And we used this very unique park as a focal point for our research, because one of the questions that I had was, how do humans mess this up? Because we mess everything up. And Halama Beach, raise your hand if you've been to Halama Beach. Sweet. Okay, if you haven't gone, you need to go. But Halama Beach is very popular, obviously, and it is alone in the middle of an otherwise pretty undeveloped stretch of coastal habitat. So as far as human activity goes, it's pretty concentrated at Halama, and it kind of diffuses out from Halama. So a lot of people there, not a lot of people on the rest of the coast. We set up cameras northwest of it and southeast of it, sort of in the same extent in either direction, to see if we could capture a signal of human disturbance when it came to mammal beach use. This is what a lot of our cameras look like. Again, they're motion trigger cameras. We place them non-randomly. Not the best science in the world, but we didn't know if there were even mammals on beaches. So we found game trails and we set up the camera traps next to those game trails to hopefully capture mammal activity. This is what a lot of our camera traps looked like. They were right up on the edge of the coastal ecotone, and the result of this is that a lot of them were on cliffs, and a lot of them got washed away during the winter storms of 2022, which is why our research ended in December 2022. I just wanted to throw this in here. This is Steph laughing at me because we put this camera in a really dumb place and it got washed away by the storms. Again, I want to shout out to all of the undergrads and postgrads and no high school students this time who helped me out with this research. I got unattractive pictures of everybody on the camera traps. So thank you so much. This is how I'm thanking you. Thank you everybody for your help, which again would not have been possible without all of the incredible field help that I've had. So my team and I would go out once a month and we would retrieve camera trap data from the field. We scanned the data for misfires and took data on images of people. So we had a signal of human activity. We uploaded the images to a really cool online platform called Wildlife Insights, which uses artificial intelligence to classify animals in your pictures to species. Pretty good. I'd give it like a B- at identifying animals to species. And then from those identifications, we extracted data on the wildlife species that were present, counts of those species, and their behavior. And then we were able to answer our first question, which is who is out there? And the answer to that question is mostly coyotes. We also saw wild pigs and deer and a whole bunch of small mammals, but over half of our sightings were of coyotes. Very, very active on beaches and so pretty. I love coyotes now. And they just make for the best pictures. They're so fun. But again, we also saw these feral pigs. They're not native to the area. They are pretty invasive, but they're very interesting. They travel in these big packs and are really spooky, especially when they roam around at night. We also saw a lot of mule deer, males, females, babies, hanging out on the beach. A lot more rodents than we expected, which I thought was very interesting. So a whole bunch of rodents on the beach, presumably doing, we're not sure what. Occasionally we caught a bobcat on the beach, which was really fun. About 3 sightings of black bears. Kevin's nodding. People who talk about black bears. When we were getting anecdotal reports of beach mammals, it was always people excited about the bear prints on the beach. And one time we caught a mountain lion, which was pinnacle of my PhD so far. So then we can answer the next question. What the heck are they doing out there? Turns out they're eating. So over 10% of the recorded behaviors, this is actually like the biggest chunk of recorded behaviors, were food related. So foraging for food, nose to the ground, like this coyote here, actively eating or carrying food. And we got several observations of marine derived food. We found mostly coyotes eating stuff, a lot of rocky intertidal invertebrates. So this coyote is holding a gumboot chitin, which if you work in the rocky intertidal, join me in wondering why they're eating this thing. I hope they're playing with it because it's called a gumboot chitin because it's literally the texture of a boot and they do not taste good, but the coyotes seem to like them. They also ate a lot of sand-dwelling crustaceans, which should be no surprise to anybody in the crab lab. Caught a lot of footage of them eating near-shore fishes, both big and small. This one is holding an adult midshipman fish in its mouth that it, I assume, pulled out of the rocky intertidal where they lay eggs and seasonally, and the males guard the nests. They also got a lot of seabirds, both scavenging and predating, pelicans especially. This is my favorite picture so far. This coyote looks like it sprouted wings because of the way it's holding this pelican. A lot of marine mammals as well. So coyotes, again, not only just scavenged marine mammals, but predated on them. And this has been corroborated by a friend of mine, but they pull the fins off of harbor seals and sea lions that are resting on the beaches, leaving the rest of the harbor seal and the sea lion alive to struggle without fins. We got a lot of footage of them holding flippers in their mouths and the skin of sea lions. This one was pulling a sea lion's entire carcass up the side of a cliff. That took it about a day, and luckily it did it right in front of my camera. It was really fun to watch. The pigs are also down there eating, which is very interesting. They are eating mostly the sand-dwelling crustaceans, and they root around in the sand just like they would in the terrestrial habitats. And they make these big sort of scour troughs with their snouts while they're looking for the invertebrates. We also saw a bunch of deer eating mostly invasive vegetation, sea rocket, ice plant, some invasive grasses. but every once in a while we would catch one actually eating algae, which was pretty cool. One of the 3 times that we saw a bear on the beach, it was holding, I think, the carcass of a sea lion or a harbor seal. We found the rest of it on the beach later. But this one was carrying this carcass home. Okay, so we answered who they are, what they're doing, and now we can ask the more complicated questions. We get to use statistics now. We asked where they're doing stuff. Before we asked where the mammals are doing stuff, we had to ask where the people are so we could see that signal. Now, people were significantly more active at Halama Beach, no surprise there, and their activity kind of sort of diffuses out as you get further from Halama. There are a couple of other minor hotspots of human activity, both which are really popular surf spots. So again, no surprise there. We predicted that generally, the further you got away from Halama Beach in either direction, the more mammals we would see. So we predicted this positive relationship between distance from Halama and the seconds of activity adjusted for effort. We took all of the data, all of the hours of monitoring that we did, and we ran some generalized linear mixed models. And that's pretty much what we saw. A kind of a significant positive relationship between distance from Halama and seconds of activity per trap night. Now, one of the other things that we predicted was that more synanthropic, which is a fancy word that means doesn't care that much about humans, the more synanthropic wildlife would be less averse to Halama. So maybe they wouldn't have this positive relationship with distance from Halama. Whereas other more frightful mammals might have that positive relationship with distance from Halama. So we thought that maybe coyotes specifically would be less averse to human activity. But it turns out they weren't. Turns out that they were just as freaked out by human activity as the other organism that we looked at, which was the deer. So next we were able to ask when are these mammals most active? And we were specifically interested in seasonality. Is there any seasonal signal? Now, it's pretty hard to measure seasonality in California where we don't really have seasons. So we looked instead at rainfall as a proxy. And on our coastline, we have basically 2 periods. We have no rain and rain. And the rain is usually about 2 inches of rain. So we were able to take, instead of like a monthly average, a 90-day average, which allowed us to kind of smooth out the signal of rain and allow time for any vegetation or anything like that on the terrestrial side to grow and for animals to respond to it. Now, what we predicted was that mammals would probably be pretty well supported by terrestrial resources in times of rain. If you think about bottom-up processes, rain makes there be more vegetation. Vegetation supports smaller mammals, and smaller mammals support bigger mammals. So we predicted this negative relationship with this mean 90-day precipitation and seconds of activity adjusted by effort. Kind of what we saw. We found a significant negative effect, but it was pretty weak, and there was a lot of noise. So again, we dug into these 2 species for whom, quite frankly, we had enough data to dig into. And we found that they're kind of doing different things. So again, deer are herbivores and coyotes are carnivores. And for whatever reason, deer seem to be responding positively to rainfall. So what that means is that there are more deer on the beach when it's been wetter over the last 90 days. And coyotes are following what we predicted they would do, and they're responding negatively to rainfall, which to me suggests that they are well supported in times when it's wet, and when it's dry, they come down on the beaches to subsidize their resources. So why does this matter? I took away the why and I decided to instead ask, how important is beach access to these mammals? And it seems that beach access is pretty important to these mammals. But unfortunately, my PhD is over and I am handing the baton to my lab mates, Grace and her undergrad Rosie, who are asking how important this beach activity is to these mammals by looking specifically at nutrient signatures in the fur and in the scat of mostly coyotes, because they're the ones who poop everywhere, to ask, like, what is the contribution of marine resources to their diet? So instead of just how much time do they spend on the beach, how much do they actually forage on the beach, and how much of their diet is comprised of marine resources? So really cool future for this work. I'm really excited to see where it goes. Okay, so to wrap up, for infectious processes, potentially absent, they might be understudied. Unclear whether or not infectious processes are missing from the rocky intertidal because the system's degraded or the habitat's naturally unsuitable. Cross-ecosystem processes are definitely playing a role in the intertidal zone. They're potentially pretty important, at least for some consumers like coyotes, and they're definitely being impacted by human activity, at least human infrastructure like campgrounds. Okay, before I wrap up, I have so many people to thank for this. Armand is right. I was operating on not very much funding, but I still have funders to thank. The Coastal Ranges Conservancy, the Nature Conservancy, the Rafe Sagarin Fund, the Space Force helped me a little bit. This department, I got the Lewis and Clark Fund, Hollister Ranch, and the Coastal Fund all contributed resources, financial or otherwise, to these projects, and was not possible without them. I have to thank my committee, my amazing committee. I'm lucky enough to have spent time in the field with all three of my committee members. Unfortunately, I only took embarrassing pictures of Armand and Hillary. Chelsea, sorry, I didn't get any embarrassing pictures of you in the field. My undergrads, you guys are amazing. Thank you so much for your help. I'm so proud of all of you. A lot of my undergrads have since gotten into grad school or are going to apply to grad school or are on some incredible upward trajectory of their lives. They're all doing amazing, amazing things. And I feel so blessed to have been part of your careers. Thank you. If your picture's not on here, count yourself lucky because I only take embarrassing photos. Again, if your picture's not on here, you're lucky. I want to thank all my friends. This department has brought me so many incredible people, so many incredible people. And everybody here is amazing. I love you all. I'm sorry I put your face on this slide. I have to thank my housemates, the doghouse. I wouldn't have done this without you, especially Sam. Sam beat me up every day to make sure I finished my dissertation. It wouldn't have gotten done without her. She's the co-author on the whole thing. I met this weird guy on the internet, and he stuck with me for some reason through the latter half of this PhD and made sure I ate every day, which is a big deal. Took really good care of me and helped me both in the rocky intertidal side of the field and the terrestrial camera trapping side of the field, and walked with me while I cried when all my cameras got washed away by the ocean. Sorry guys, I'm gonna make you cry. Mom and Dad, thank you. You guys, damn it. You guys encouraged me from day one, literally 3 years old, was like, I'm gonna be a biologist. And my mom was like, let's look up the word for that kind of biologist. And then, 20 years later, we're in the field in waders. I have some excellent pictures of my dad in waders helping me with my research. I am so incredibly lucky. You guys are great, but none of you are as great as Juniper. I love you all. She is the best field assistant, and yeah, you. She had to check every single organism that we pulled out of the tide pools to just to make sure. And she deserves an honorary PhD. And with that, I will take questions.
Fantastic talk.
Thanks.
Excellent presenter as well. I'm interested in really old human communities that would have been living alongside in tidal zones.
Mhm.
Humans, of course, can host these complex parasites. And even if people weren't themselves defecating on the beaches,
Mhm.
you all the river, and then, things are being washed in the ocean.
Totally.
Could just sustainable human population be missing from the, of course, with all the water quality stuff, we're not letting any of that stuff on the ocean, but they're eating more or less of it. Anyway, yeah. So
Yeah, I actually hadn't thought of that. We can be hosts for some nematodes, but I don't know. There's tapeworms that we can be hosts for, but I don't know. That's a really interesting question. I think we would have to look in middens and see if there's coprolites, fossilized poop, that I mean, can Metacercaria fossilize? So I don't know. That's a really interesting question. It's possible. It's possible. Yeah. I know. Yeah, so one of the things that I really wanted to do was use some eDNA data that Mary McElroy collected to make kind of like mini food webs for the California coast and see if this like hypothetical simplification of food webs that we assume is happening is actually happening. So like take that eDNA data, apply it to the food webs and see sort of like our food chains shortening, our food webs getting less complex. So that's something that Mary and I have talked about doing in the future. She actually just emailed me yesterday. So that could be a very cool potential application. And I'll let you know in like 8 months when I attempt to apply this to government work. Yeah, Jordan. Oh God. I think sea level rise is actually less of a problem than like storm severity and like periodicity increasing because over the last just like 15 years of living here in Santa Barbara, I've seen our intertidal areas just get less and less biodiverse, and it seems it's because they're being buried in sand for longer and then scoured out by that sand for longer. So for those of you who don't know, one of the processes that happens in the intertidal, especially in California, is we have this like sand inundation, usually in the late summer as the waves kind of slow down. And that's when we get like nice big beaches and everyone's like, yay, beaches. And then in the winter, we have all this storm activity that then scours out all of that sand and the sand gets taken away. And that's when the rocks are exposed, but it also like literally wipes creatures off of the rocks. So I think that more than sea level rise, it's the storms that are really going to mess stuff up for the Rocky Intertidal. It's going to change the way that that works seasonally. Yeah, Robert. Totally. Yeah, I had a whole rant about this, but Armin told me to take it out. um, I'm pretty worried about them, honestly. Like, let me see if I can go back to the slide, but the slide where there's all of the like pig scouring, whatever, we'll just leave it. They do that for like hundreds of meters. So they'll go out in these big groups of like 20 strong and they reproduce every year and they have no predators pretty much. I mean, they have predators, but not that many on the beaches. And they reproduce, each sow has like 10 babies. And so they grow in these gigantic groups and they just root through the kelp rack. And as they're doing that, they're eating the kelp rack. And I assume they're also in there for the like telitrid amphipods and the sand crabs and stuff like that. And those organisms are really important for the sandy beach food web. Like they are essential processors of nutrients. They take the kelp and they turn it from trash into sand nutrients, basically. And if the pigs are putting a dent in that population, they're disrupting a really important ecosystem function because that like those kelp nutrients go into the sand and they feed dune grass and like without them there is no beach vegetation. So pigs could be really messing stuff up, but I mean I have no idea. It's entirely possible. Hey, see, I'll go there.
So what about other cases? I don't think that they could be having any impact on the parasite diversity.
Yeah, this is something I talk about when I teach parasitology about how like invasive organisms usually leave their parasites behind, and that's part of why they're so successful. Further north, there are issues with the green crab, the European green crab being invasive in the Rocky Intertidal. And one of the reasons it's so successful is because in its home range, it has this parasitic barnacle that shrinks it. So it gets smaller because a lot of its energy is going towards the parasitic barnacle and not towards growth. And here it doesn't have that parasitic barnacle, could get a lot bigger and it can outcompete stuff. So, and it also doesn't have parasites. So like, that's another way that the ecosystem could be suffering. I mean, I don't know if we would see that on the food web scale, but that is a process that we're aware of that's happening. Lisa. So I was thinking of like, there are some nematodes that can go from having like just 2 hosts to like 5 hosts in like within the same species, but no. On an evolutionary scale, it's possible, you know, like not within a population necessarily, but on an evolutionary scale, something that I would think on the order of like millions of years, something that would have had a more complex life cycle might have evolved to have a more simple life cycle. But what probably happened is things that had complex life cycles just aren't making it there. That makes sense. Yeah. Okay, Sam, and then Julia.
I thought it was really interesting when you mentioned that there's a study showing that zonation showed greater parasitism or parasitic interactions in the high intertitle zone. And I was wondering if it's possible to, or if there are obstacles to you like categorizing the zonation or mobility of species in your food web and seeing if that plays out in your own food web.
Yeah, that's definitely something I explored kind of like a couple of years ago and then sort of abandoned because on the food web scale, it was a little bit difficult because a lot of things are either sessile or they're highly mobile. So things are either moving between all the zones or they're stuck in one. It's definitely something that I've been wanting to talk about my friend who's at UC San Diego about. But yeah, it would be really interesting to see if like things that are zoned, that are stuck in like higher intertidal zones have fewer or more parasites than in other zones. Yeah, but honestly, characterizing the zones in itself is a whole boondoggle, so maybe for another day. Yeah.
Great talk, is awesome. This is maybe kind of a basic question. How do you know, like, found all the parasites in the section? Like, are there really good guidebooks to get them down the species?
No. I did my best. Yeah, the answer to that is math that I was that I ran out of time to do. Heaven. Basically there are like statistical ways to see what the potential is that you've missed a lot of parasites. In the actual like dissection in the moment, there are protocols that we in our lab follow to do like whole fish, whole crabs, whole worms and stuff like that. So I followed a lot of what Dana Morton set in place. So she's the one who was the author of the kelp forest food web, and she did a bunch of dissections of organisms that are very similar to mine. And I basically just followed her procedure. But you take, if it's a small enough animal and it doesn't have a hard exoskeleton or isn't a fish, you can kind of do a whole squash and then look for stuff, which is it sounds weird if you aren't me, and who's been doing whole squashes since I was, 19. But yeah, you basically squish the whole thing. Or you can take out every single organ and squish the whole thing. And that's kind of the best way to make sure you've got everything. Yeah. Yeah. Oh, Renata. Renata, do you actually have a question?
I do have a question. Also, congrats from Seattle. We're here cheering you on. But I did have a question. You talked earlier in your first chapter about how you put a lot more effort into having a highly resolved food web. Is that standard across other ecosystems or disciplines, or do you think that is really valuable work that will be applied to more ecosystems in the future?
Yeah, that's a great question. This is my friend who's an engineer for Microsoft. Yeah, so that's a great question. It is something that sort of as in the food web community, we're hoping continues to be the standard. Like in the past, it was sort of more accepted to lump things together into functional groups, but in the last 2 decades or so, people have really been trying to progress forward with resolving things down to species because biodiversity is really important. And it's also really important to understand things like functional redundancy. So like, let's say you have 30 species that all do the exact same thing. They're all eaten by the same predator and they all eat the same prey. It's important to know that there are 30 of them. You can't just lump them all into one big group just because they do the same thing. So I'm hoping, and I think a lot of other people who build food webs are hoping that they will continue to be resolved to species wherever possible. It's not always possible because people from 1916, you know, publish papers that say birds eat crabs, and you're like, this is not helpful. So, you know, we do our best.
We have one more question from Seattle.
Oh, good.
Can you tell me what Juniper's favorite creature to eat out of the intertidal is, please?
Crabs. She likes crabs. Oh, no, that's not true. She likes the sand isopods. Those are her favorite. Okay. Yeah, Gabe. Thanks. Yeah. That's a really good question. I don't know about the last resort question. For the coyotes, I don't think so. I think that the beach coyotes know what they're doing. I think that, like, if they smell that there is a dying sea lion on the beach, they'll come eat it. It's not a last resort. Like, they want that. For the deer, I'm not sure. I honestly have no idea why the deer are down there. Like, it does not make any sense to me. They got way better stuff to eat elsewhere, even if it's in the middle of summertime. So I don't know why they're down there. I looked for evidence of avoidance, and I did not find it statistically. I have a feeling that the pigs are driving things off, but I haven't found it statistically. I'll let you know at the end of the Hollister Ranch project when I have more data. Armand.
So, do you think that the high proportion of filter feeders in the Rocky intertidal zone affects transmission of parasites?
Yes, definitely. So that wasn't something that I included in the like features of the Rocky intertidal that might be hostile to parasite transmission. But like I showed you when we broke things down into functional groups, 30% of the organisms in the Rocky intertidal are filter feeders. That's a lot of vacuuming that's happening. So a lot of parasites propagate and transmit by releasing infectious stages that move in the water, even the terrestrial ones. And they're all like the size and shape of zooplankton. They are zooplankton, essentially. And those filter feeders are a lot of times evolved to eat zooplankton. So there's been some evidence in other research done in our Rocky intertidal zone right here at Campus Point that anemones can actually depress infections between barnacles by consuming the larvae of the barnacles parasite. Yes, Athena.
I was just wondering why you said that there were a lot of rodents, but you didn't touch on that.
Yeah, Athena. So basically our cameras were not designed to pick up rodents. So I felt it was a little inappropriate to statistically analyze the rodents on their own because basically our cameras were all at about knee height and rodents are at ankle height. And so the only time we really got like a lot of rodent data was when the cameras fell down. So we would have seen a signal of like, oh, there's a really positive relationship between rodents and rain because every time it rained a bunch, the cameras fell down and we saw all the rodents. So thank you for reminding me of that. Yeah. Yeah, that's the next step. If I was to stay in academia, which I'm not doing, that's what I would do next. So I started that project with an undergrad a couple of years ago to sort of see if we could find a signal of marine parasites in terrestrial coastal mammals. And we didn't make it that far, but we found a lot of parasites. We just hadn't classified them to like marine or terrestrial yet. There is some research, again, from right here at Campus Point by Sarah Weinstein a couple of years ago that suggests that raccoons, when they're foraging in the intertidal, actually end up with marine-derived parasites. So yes, I guess is the short answer, and it would be really cool to continue to explore that. So yeah. I'll take one more because we're like way over time. or no more. Okay, thanks, guys. Universal. Thanks, Joey.
That was a masterclass
Oh.
on how to do a presentation.
Thank you.
That was just beautiful.
Oh, thank you so much. Thanks,
And
Jess.
it's great fun for me.
Thank you. I'm so glad that you got here.
Oh my God.
Yeah, that's fine. I'm glad you're here. Thank you. So what do we do? I know you guys said Chelsea's still here.
Right. All you had to do was do.
Okay.
Could have like, I don't know, sat up here and, fluffed terribly and it's fine.
Yeah.
So the process, you did it. Yeah. But I usually, like hang out and talk.
Yeah.
And by the way, congrats on the job.
Thank you.
What is it that you got?
It's the Rocky International. Yeah. 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 So, I'm really excited. I start on January 13th. I get just a long month off. Yeah, it was start date and everything. Okay, I got it. Chelsea, do you have anything for me? I gotta turn you up.
I got plenty of questions. Are we sitting down in a closed door after the fact or no?
We can sit down for a minute and talk where it's more of like a formality than like a real thing, but
Mhm.
I thought that you had to go.
Nope, I'm good.
Oh, excellent. Okay, fantastic. Yeah, let's have a chat. Hold on, I'm gonna
Take your time.
I'm just gonna stop the recording.